You see, oh, 7 repeats, and a few more, oh, another 7 repeats of the same thing, another 7 repeats, and you spot those, good indication. Okay? And 1 and 4 tend to be similar. Okay? Like 2 and 3 tend to be similar in their R group characterization. Okay? So again, because we have 2 alpha helices that go side by side, that makes the interior, like those A and D portions right here, tends to make them probably nonpolar. Why might they be nonpolar? Because if these two are facing each other, what's in the middle here? Well, what's not in the middle here? Water. Okay? Nonpolar near nonpolar. Okay? Well, what's on the outside? What's out here around C and B and F and all this good stuff? What's on the outside there? Water. So what do those tend to be? Polar. Okay? So if you look, if we talk about what's the secondary structure, we're kind of skipping ahead here, but what's the secondary structure of alpha keratin? Alpha helix, that's it. Okay? So for tertiary structure, what would you say? Remember, tertiary structure is not looking at these two, we're just looking at one piece. You have to tell me about what the positions of the R groups look like. So the R groups here produce what type of helix? One side that's water loving and one side that's water hating, right? What's that called? Antipathic helix. Everybody okay with that? You notice, do I put on tertiary structure coiled coil? No, because a coiled coil are interactions of 2 different polypeptide chains, which is what? Quaternary structure. Everybody okay with that? Please do not tell me your tertiary structure is coiled coil. It is not. Tertiary structures is looking at one particular amino acid sequence. Okay? Here's the other thing, though, about quaternary structure. Alpha keratin tends to contain, in addition to the heptide repeat, tends to contain lots of cysteine residues. What's the R group on cysteine have in it again? Sulfur. Okay. We talked about sulfur. Sulfur, you can actually bring 2 sulfurs together, get rid of some hydrogens. What type of reaction again? You're losing hydrogens, gaining hydrogens. What's that called in biochemistry? Redox. Redox? Anybody remember? Oxidation reduction reactions? Yeah? Okay. When you do that, when you take 2 cysteines and you bring them together, you lose those H's. What type of bond is that called? 2 sulfurs coming together. Disulfide. Okay? Now you have disulfide interactions between those 2 different chains. So what type of which one of these is that? Different chains. Quaternary structure. You also have disulfides. Do you think that makes this coiled coil stronger or weaker? Stronger. Remember, disulfides are kind of unique. They're actually physical bonds between these 2 things. Do you think, based on all of these things, you have a repetitive? You only have an alpha helix, really, for the most part. You have an antipathic helix, so those want to stay together. And you have disulfide bonds, which hold them together. And you have a coiled coil, which basically is left-handed and holds them together even tighter. What do you think about the stability of alpha keratin? Very stable. All of these things contribute to the stability of alpha keratin. Okay? Here's the thing. Alpha keratin, what it looks like, and what overall we see with it depends upon actually that cysteine count, or the percentage of cysteine that we have in it. Okay? The cysteine content that we have actually directly is related to the different properties of keratin, for alpha keratin. Okay? The more cysteine you have, what do you think, stronger or weaker? More stable, less stable? Stronger, more stable. Okay? Because you assume that the more cysteines you have, the more what you have? Disulfide. Because you link those 2 coiled coils, those 2 alpha helices together, even more. Okay? So that's why, and again, the more that you have, you get more alpha helices. So, for example, this is in the 80s, but perm kits were really the thing in the 80s. I know, firsthand. Anyway, they smell like really bad. Anybody ever had your mom or no? Okay, all right. It's fun when your child's young or really young and they can't really complain. But they smell like sulfur, right? Anybody smelled them before? Yes. Yeah. Why do they contain sulfur? Because if you increase sulfur, guess what you probably can increase? Disulfide bonds to hold those curls and things together. Okay? It increases the twisting that you can have. can actually look, if you wanted to analyze different hair types, you could actually, take the alpha keratin that's in your hair and compare it to somebody else with a different hair type, run through a mass spec and figure out what's the cysteine content. And I can pretty much guarantee you that those with naturally curly hair have what? More or less cysteine than those with straight hair. More. Okay. If you wanted to, though, you can also have beta keratin. So beta keratin basically is an extended conformation of alpha keratin. So if you took the two ends of alpha keratin and you pulled it apart and you broke all those hydrogen bond interactions, it would naturally probably settle into a beta sheet conformation because it's a repetitive structure. Okay, it wants some type of secondary structure. It doesn't want to just be nothing. Okay. Questions for me on the 4 levels of protein structure of alpha keratin?
Can you explain the beta sheet? Just the last part.
Sure. Well, the beta sheet, remember you have to have 2 different beta sheets, right, coming together. So what you do is if you pull this apart, if you pull each of those helices apart, you end up getting 2 kind of beta sheets, or I don't know if they exactly look just the other way, but so you have, this inside or whatever, where your R residues point on the inside would be your nonpolar, and then out here would be your polar. Yeah. Does that kind of help visualize? Yeah. So it's basically, if you remember, the hydrogen bonding network in an alpha helix is close or far apart?
Close.
Close. Beta sheets, right? It's between those 2 different residues that you get that interaction. Does that help a little bit visualize? Yeah.
What type of fibrous protein would elastin be?
We're not going to talk about elastin. What do you mean type of fibrous protein?
Well, I don't think, considering it had collagen is more of an alpha, I'm assuming considering the elastin has more of a give to it, wouldn't it be a different kind of fibrous protein than the alpha keratin?
I don't know what you mean by different.
Well,
So you mean like different secondary structure, different tertiary structure? It's all going to have different, they're all going to have slightly different secondary, tertiary, and quaternary structures. Every single protein is going to.
Yeah.
Now, just because alpha keratin is dominated by an alpha helix and collagen is dominated by alpha helix doesn't mean there's partially random coils in there, depending upon the environment that it's in. If you think about it, if you think about your nails versus your hair, right, are they different? Yeah, so do you think the keratin content of them might be slightly different in terms of their structure? Same thing. Elastin, when you put it, and same thing with elastin, similar but different, if you put it under different conditions and different environments, it can adopt different properties. You have to have proteins that can adapt to the different conditions that they're in.
Okay.
If you don't, you're gonna be dead. I don't know how else to put it. I was like, you can't live if you don't have proteins that just kind of sit around and can't really do anything. Yeah. I know that doesn't really answer your question, but for now. Yeah.
So what you have on the board, is that the tertiary structure of the beta sheet?
It would be a beta, yeah, for an example of beta keratin. Yep. So it would be, you'd also still, if this were beta keratin, repetitive beta sheet, amphipathic sheet, and then you would have, you can still have disulfides that exist there, but you don't have a coil anymore. But you have beta sheets that hydrogen bond.
What do you mean by this category?
So the primary structure, right? So what's the primary structure again? So if you had something like GST, G over and over and over again, right? You see that repeat, yeah. So again, you have the 1 and 4 with the alpha keratin. Good question. Other questions for me? Because we're going to leave keratin and talk about collagen, which is similar, but a little bit different. So collagen, over here, okay, is a triple helix. So, never mind if I already saw our beta sheet here. Okay. So if you look at collagen, collagen is also a fibrous protein. Okay? It's also a fibrous protein, which means its primary structure is also probably repetitive. Okay? Here's the thing, collagen's a triple helix, so you can probably tell me already something about its quaternary structure. But collagen's actually really found in what we call harder tissues. So again, if you had to compare your bones and your teeth to the strength of your hair or your nails, which one's stronger? Your bones and your teeth, right? So what's the stronger overall protein, you think? Collagen or alpha keratin? Collagen. So now we're going to talk about why that's the case. Okay? If you notice, the structure of collagen, do you think it's repetitive just like alpha keratin? Do you think it's mostly helical just like alpha keratin? Think it has a coiled coil just like alpha keratin?
Yes.
Yes, but there are slight differences. And those differences might be subtle. For instance, instead of just 2 coils, we actually have 3. But those are enough to give you massively different properties of collagen versus alpha keratin. Okay? Here's the thing. The primary sequence of collagen, here's why it's easy to remember, is that collagen's a triple helix. The repetitive sequence tends to be in 3s. Okay? So here's the thing. Does this mean it only repeats in triplets of 3 of these 3? No. But you might occasionally see these repeating over and over and over. Okay? That repeating triplet is glycine. What do we know about glycine's R group? Tiny. Okay? So if you have a lot of R groups that are really tiny, do you think things can pack better or pack worse? Better. If things can pack better, what do you think about its stability? More stable. Everybody okay so far? So that's already one thing going for it. Okay? Here's the other thing. X is oftentimes proline. Do we remember what's so special about proline? Right, it's got its R group in the, I'm assuming this is R group is in the backbone, or the R group's in the actual backbone there, makes it more conformationally rigid. So that means if you have something that's more conformationally rigid, and you're trying to make something that's stiff, you think it contributes to the stability or goes against the stability? Contributes, okay? Y is hydroxyproline. How do you think proline and hydroxyproline differ? An OH group, a hydroxy group, okay? There are enzymes that put, remember when I said we can take amino acids and we can modify them? Right, this is an example. You can take a proline, you can add an OH group on it. Okay, and it'll give it a slightly different properties. Because if anybody remember from your amino acid assignment, what type of residue is proline? Polar, nonpolar, acidic, basic, what? Nonpolar. What happens if you put a hydroxy group on it? Turns into something that's polar. Everybody see that? Okay? It still confers that structural rigidity, but now you've got slightly different properties. Okay? So if you were proline, would you want to be in the middle of this helix or facing the outside of the helix? In the middle. If you're hydroxyproline, where do you probably want to be? Outside. Everybody okay with that? You see the difference? Just that addition of that hydroxy group gives you vastly different properties, enough that it can actually change the overall structure of a protein. Okay? All right. So now, we just saw, for collagen, what's our primary sequence? Repetitive. So here, repetitive, I would put for alpha keratin. It's important now to distinguish the difference. Usually with alpha keratin, we see repeats of, what do we see repeats of? 3. And it's glycine, proline, and I'm just going to denote that as proline OH, or hydroxy. Okay? All right. So remember, hydroxyproline, or hydroxyprolose for another name for it, is basically just we put OH groups on proline. Okay? Now there are different places where you can put those OH groups because you have several carbons that are accessible. Okay? You're not going to put it, so again, mostly what you're making, pretty much the only things you're making, because there's only 2 possible ones right here because you have the peptide bond forming. You have 3 hydroxyproline and 4 hydroxyproline. And the 3 and 4 just denote what again? The position of the OH. Okay? It doesn't matter to me. Okay? So, we take proline and we stick an OH group on it. Do you think that spontaneously happens all by itself?
Yes.
No. You need something to help it along. Okay? This is where those enzymes come into play. Okay? So we're going to take proline, which is already in the amino acid sequence, and we're going to have another enzyme come along, and we're going to modify some of those prolines, which are already existing on there, and change it to hydroxyprolines. Okay? It's a modification. It's a post-translational modification. After you've made the protein off the ribosome, you're going to do this. Okay? And guess what? How do you know something is an enzyme? It ends with ACE. And usually the beginning phrases tell you what it does. So again, prolyl hydroxylase. What do you think this enzyme does? Puts hydroxy groups on proline. Okay? I know. The way that we name enzymes is insane. Okay. So here's the thing though. This enzyme requires ascorbic acid or vitamin C to do its job. Okay? So if you're vitamin C deficient, you don't have any, are you going to make any hydroxyproline?